and go through the appendages of the skin. Some of those you already looked at in the lab that we were doing today. So we're going to talk a little bit about their structures and what you need to know about them for the test. So all of the glands that are found on the cutaneous or the skin are exocrine glands, which remember means that there is a duct that allows them to release their fluid to the surface. You have two different types of exocrine glands, sebaceous and sweat. Sebaceous is your oil glands. You also have hair follicles, and nails. So let's talk about the oil or sebaceous gland. They produce oil, which is also called sebum, and you do need to know that term. Its function is it provides lubricant for the skin, it prevents brittle hair, and it also kills bacteria. So when it's talking about lubrication for the skin and preventing brittle hair, remember the sebaceous glands are located on the hair follicle, and they release oil into the follicle and onto the hair shaft as it's coming up. Most have ducts that empty into the hair follicles. Other will open directly into the skin. Glands are activated at puberty. So it is the gland that is sitting there on the hair shaft. Here's an enlarged picture of it. Here is your hair follicle. Here are the sebaceous glands are up towards the top. The secretory glands are down below. Your sweat glands are also called your sordiferous glands, as well as sweat. They produce sweat, and those are widely distributed around the skin. Some of those are found on the palms of the hands and the soles of the feet, which is what we were going to do with the iodine to find that. For your sordiferous glands or your sweat glands, there are two different types. They are eccrine glands and apocrine glands. The eccrine glands open to pores on the surface. So the eccrine glands produce sweat, which is clear. The apocrine glands are ducts that empty directly into the hair follicles. This begins to function at puberty. They release sweat that also contains fatty acids and proteins. Sweat is basically your sordiferous glands. Oops, I went backwards on that one. I was wondering why that seemed like a repeat. So the apocrine glands release sweaty, it has fatty acids. That's what you find usually under the armpits. It produces more of a substance that kind of smells. This was a picture that you saw on your lab. Then an eccrine gland duct in there. It looks like the cuboidal, the dermal tissue, etc. When you look at those, for sweat, its composition is mostly water. You do have some salts and vitamin C. We talked about the fact that you do release some urine and urea in your sweat, so it does release metabolic waste. It releases fatty acids and proteins. That's only the apocrine glands. The function of the sweat gland, it helps to get rid of heat, helps to get rid of excrete waste products. It is acidic, and that inhibits bacteria growth. So we talked about the fact that natural substances on the skin help to, although we have bacteria all the time, helps to reduce the amount of bacteria and bacterial growth. Odor that we have from sweat is associated from bacterial growth. Hair is produced by the hair follicles. It consists of hard keratinized epithelial cells. Melanocytes provide the pigment for the hair color, so the same melanocytes that are found in the basal layer of the cell are also found in the hair follicle. Hair grows in the matrix of the hair bulb, and just like the skin layers, the basal layer is the one that it grows out from as it grows and divides and pushes up into the hair shaft. The cells die, and they are no longer living cells. Once that basal layer dies off, then the hair dies. So here's a hair follicle. You've got dermal sheath, you've got epidermis. Melanocytes are in there. That is the basal layer on the most inside. You've got connective tissue and blood vessels that are in the middle of there that feeds those cells that are in the basal layer. And then you've got the subcutaneous or the fat underneath. Hair anatomy, it's got the central medulla, and we saw that in the picture I drew on the board. Sometimes you can see a line in the medulla, sometimes you cannot. Cortex surrounds the medulla, and then the cuticle is on the outside. The cuticle of the hair does have scales on it with different patterns. Cuticle is the most heavily keratinized because it's waterproofing it, it's protecting the hair. When the cuticle itself is damaged, then that's when you have split ends, etc., and the hair doesn't stay in its normal shape. With the hair follicle, you have a dermal and epidermal sheath around the root. You do have an erector pili muscle, and that is smooth muscle. Its job is to pull the hair upright when you're cold or frigid. You have sebaceous glands and sudoriferous glands. Sebaceous glands are the oil glands, remember. The sudoriferous glands are the eccrine and the apocrine glands. So when you look at the hair shaft there, you've got the sebaceous gland, you've got the shaft coming out. The bottom part here is the bulb of the follicle. Above that is the hair root. There is the muscle called the erector pili. So there are the scales magnified, and they overlap one another, and part of what they do on the cuticle is they help to protect the hair. On your nails, it is similar to the hair. It is a modification of the epidermis. It is also heavily keratinized. The stratum basal is below right beneath your nail bed, and that's where the growth occurs. The fact that it lacks pigment makes them colorless, or makes it pink in nature. The nail structure, it has the free edge that extends over the finger. The body is a visible attached portion. The root of the nail is embedded underneath the skin, and the cuticle is the proximal nail fold that projects into the nail body. So your cuticle is at your proximal nail fold. I think, yeah, here we go, the picture is next. So your cuticle is your proximal nail fold, and the cuticle comes off of that. The root of the nail and the nail matrix is there. The nail bed is down at the bottom, and that's where you have the cells that reproduce back in the nail fold and in the nail matrix, and the free nail is up above it. So you've like the hair and like the layers of the skin is it what gets pushed out and growing and extending once it is beyond the fingertip and into the free edge, no longer has a blood supply and is not living cells. All right, we're going to talk a little bit about burns and a little bit about melanoma, but you guys are going to be doing some projects that will deal with it. When somebody gets burned and the tissue is damaged and the cells die, it can be caused by heat, electricity, radiation, or chemicals. Some big health issues they have when they have burns are dehydration, electrolyte imbalance, and circulatory shock. And the reason for that is when there is a burn, a lot of times there's oozing, which means that plasma is coming out of the cells, and you're losing bodily fluid through those areas. One of the ways that they determine the extent of the burn is the body is divided into 9 areas, and those areas give you an estimation. Each area represents 9% of the total body temperature, body surface area. So it's the rule of nines. 9% is for each of those areas, and how many areas will tell you the total amount. So the anterior and posterior head and neck is 9%. The limbs, upper limbs is 18% anterior and posterior. Anterior and posterior trunk is 36%. Anterior and posterior limbs are 36%. First degree burns, only the epidermis is damaged. Your skin is red and swollen. Second degree, it is the epidermis and the upper dermis are damaged. It's red and blistered. Third degree destroys the entire skin layer. The burned area is painless because there is no more living nerve tissue in there, and the burn is gray, white, or black. So that shows you some of those. Burns are critical if they're over 25% of your body. Has second degree burns. If over 10% has third degree. If there are third degree burns on the hands, face, or feet. So we're not going to go through all of these because you're going to do these lovelies in some of your presentations. But one thing I did want to talk about was skin cancer. There are different types. There's basal cell carcinoma, there's squamous cell carcinoma, and then you get to the malignant melanoma. Malignant melanoma is one of the most deadly types of skin cancer. It's a cancer of your melanocytes and metastasizes rapidly into your lymph nodes and your blood vessels. And the ABCD rule is how it's detected. A lot of times when you look at a mole, that is the starting point for melanoma. So if you have a dark spot or a mole that begins to change, then you should be aware and have it checked out. So the ABCD rule is if you have a mole and it has asymmetry, two sides of the mole do not match. B is border irregularity. The borders of the mole are not smooth. C is the color. You've got different colors in pigmented areas. And then D is diameter. If the spot is larger than 6 millimeters in diameter, also if it has changed. So there's a picture of melanoma. Okay, so that is the end of the notes. We will go through these in class, but you might start to do the review sheet and some of those that go through it. Study for the quiz that is tomorrow. We will have the test over the whole unit on Thursday of next week to end out the quarter.